Inside the cell is a complex known as the degradation complex. It is made up of proteins including Axin, Disheveled, and APC. Glycogen synthase kinase 3, or GSK3, is also included in this complex. Wnt stands for Wingless-related integration site. It is a protein that can interact with the cell externally. From the outside of the cell, Wnt can communicate a signal to affect the degradation complex. This will ultimately involve the cell nucleus and changes to gene expression. When Wnt does not yet reach the cell, the degradation complex is constantly removing a factor known as β-Catenin from the interior of the cell. In the complex, one of GSK3's main role is to phosphorylate β-Catenin. This flags the cofactor to be tagged by a protein called ubiquitin. Ubiquitin tagging directs β-Catenin to a transport proteasome for degradation. This prevents β-Catenin from accumulating in the cell. The presence of β-Catenin in the nucleus is a major feature of this signaling pathway.
Within the nucleus, a cofactor known as TCF represses genes. Without the presence of β-Catenin, TCF continues to act as a gene repressor. TCF would need to be inhibited by β-Catenin present in the nucleus for these genes to be expressed. Wnt can interact with two transmembrane receptor proteins called frizzled and LRP5 and 6. With the presence of Wnt, the separated proteins come together and form a complex at the membrane. After this complex forms, it can alter the performance of the degradation complex within the cell. Following the formation of the complex between frizzled and LRP, the entire degradation complex now moves to the same location of the membrane on the interior side. LRP will then interact with the degradation complex by binding to Axin and GSK3. Frizzled also binds to Disheveled and activates it. When Disheveled is activated, the degradation complex becomes inhibited. This prevents the complex from phosphorylating β-Catenin. As a result, β-Catenin no longer becomes ubiquitinated and sent to a proteasome for removal. Instead, β-Catenin becomes stabilized and can now accumulate within the cell. This change to the degradation complex alters the fate of β-Catenin and ultimately controls gene expression.
As β-Catenin accumulates in the cell, it is redirected towards the nucleus. The introduction of β-Catenin in the nucleus can affect the factors that inhibit gene expression. Β-Catenin can now bind to one of these factors, that is TCF, and form a complex. This complex converts TCF from a repressor into an activator. The genes that were originally blocked by TCF can now be activated. To summarize this pathway briefly, first VNT forms a complex between frizzled and RLP. A protein called Disheveled ultimately leads to β-Catenin moving into the nucleus to allow the expression of various genes. From the external VNT signal, genes within the cells can be completely altered.